now we're going to be doing questions 8, 9, 10, which deal with a different mode of inheritance slightly. So for number 8, so sorry, number 8 reads as follows. So spotted tribbles have colored spots on a white background. For example, a blue spotted tribble carries the blue allele, which is what gives its spots the color blue, but also carries another allele, A, that codes for having spotted fur, not just solid blue fur. When two blue spotted triples are crossed, 9/16 are blue spotted, 3/16 are black spotted, 3/16 are solid blue, and 1/16 are solid black. So for this one, what are the dominant characteristics? So, if you look at the offspring that were produced in this cross, you'll see that 9/16, 3/16, 3/16, 1/16, that is our 9 to 3 to 3 to 1 dihybrid cross, or 9 to 3 to 3 to 1 phenotypic ratio that you would see characteristic in a dihybrid cross. So for here, for here with number 8, what are the dominant characteristics? Well, since 9/16 are blue and spotted, those would be the two dominant characteristics. So the dominant characteristics are both blue, which we'll have as a capital B, and it says, but also carries another allele, A, that codes for having spotted fur. So we'll say that spotted is the capital A. The next question says, what are the genotypes of the parents in this cross? Since we know that because it's the 9 to 3 to 3 to 1 ratio, we're crossing two dihybrids. So for the next answer, we know that we're crossing capital B, lowercase b, capital A, lowercase a, with another dihybrid. So that's the answer to number 8. All right, so now number 9. So for number 9, it says a white tribble is crossed with a solid red tribble to give the following offspring. We have 50% solid red and 50% spotted red. How could this happen? Hint, no mutations occurred after analyzing their DNA. Also, carefully rereads the info given in number 8 to figure this out. So, we cross two parents. So, going to number 9. What we see being crossed here is a white tribble, and we're crossing the white tribble with a solid red. So we're crossing something that doesn't appear to have any spots, but despite that, what we do obtain in our next generation is 50% solid reds and then 50% spotted red. So the question is, how can that be? Well, if the spotted allele came, and the key point here is that there were no mutations, the spotted allele was there. So solid is recessive, right? So spotted is capital A, but then the solid is recessive. So here, if this was solid reds, we know that's lowercase a because it's solid. But we have this white tribble here. So how is that possible to have a spotted tribble here that would then have to get the capital A? Well, quite simply here, this had to have had, this white tribble had to have been a spotted tribble, yet also be white at the same time. So basically, it would have white spots on a white background. So, going to the answer. So which parent passed down the spotted allele? The answer to that would be the white parent. And then the next question asks, explain how you know this is true. So the way that we know that this is true is that the white parent, had the spotted allele. So the white parent had the spotted allele, but the white spots were were not. Just thinking about how I could make this easier for you to see. Okay. But the white spots were not distinguishable on the white backgrounds. So I'll read that again. I can understand if that writing is hard to read. So I wrote that the white parent had the spotted allele, but the white spots were not distinguishable on the white background. So that's how that was able to be passed down. Okay, so for number 10 here. Okay, so 10 reads as follows. Okay, so a red tribble is crossed with a white. So 10 reads as follows. A red tribble is crossed with a white tribble to produce 100% pink tribbles. As you know, fur color is not controlled by incomplete dominance. So what is happening? Scientists analyze the chromosomes on the pink tribbles to find the presence of another allele, which they named capital D. 2 copies of the capital D allele were found in the white tribble parent, but capital D was not found at all in the red tribble parent. 1 copy of big D is found in pink tribbles. So there's actually a couple things going on. So what do you think is happening? So if you keep in mind the thing that happened with the labs, there was, at least in the labs, something that controls the distribution of pigment. So for our labs, a lab could have the genotype big B to be black, but if it had 2 of the recessive E's, that black pigment would not even be produced. Or we could have had what should be a chocolate lab or a brown lab, but even still, if it has the 2 recessive E's, that pigment is still not produced. So this is actually an example here of epistatic pattern of inheritance. So what do you think is happening? All right, so here for number 10, okay, so for number 10, what do you think it is that's happening? So D controls the amount of pigment produced. So D controls the amount of pigment produced, period. And in parentheses, I'm going to write epistasis, which is what, again, we had learned about in terms of the chocolate labs, black labs, yellow labs, as well as with the mice fur color. And additionally, this is the other thing, is that capital D is incompletely capital D, is incompletely dominant over lowercase d. So, what do we mean by that? So, what we mean is that if something has big D, that means that the full amount of pigment is produced. Full pigment. So an example would be if we have big B, big B, big D, this would be a tribble that is blue, but is fully blue. If we have big D, little d, that's going to be something where there's a partial amount of the pigment produced. So we'll say partial pigment. And if we go here and we put big B, big D, little d, so the tribble has the genotype big B, so it should be blue, but there's not as much pigment that's produced, so it would be a lighter shade of blue. And then if it's two lowercase d's, no pigment whatsoever is produced. And if we carry that example over here, if we have big B, and little d, then this would just be a white tribble. If you think about it, let's do another example. Let's say we have BR, big D, little d. So BR would be for a purple tribble. Big D, little d would be for pale color. So this would be like a pale purple tribble. So that's the idea and the pattern that's involved. So that's for number 10. Okay? So now for number 11. Number 11 says to show a cross between a pale green tribble and a pale purple tribble. So give a phenotypic ratio of the offspring below. So for number 11, here are the parents that we're crossing. A pale green tribble, so first of all, the color is green. So that's why we're going to be crossing B and Y. But it's pale in color, so epistatically, right, that should be big D, little d. And then it's being crossed with a pale purple tribble. So we're crossing that with a purple, which is BR, but pale in color, big D, little d. So let me just pause this while I set it up. So, and just like that, I set up the Punnett square. So I think that the new and tricky thing for us was to determine the new genotypes of the parents for these triples and then interpreting it. So it's business as usual for us. So foil, big B, big D, here. Outer, big B, little d, here. Inner, big Y, big D, there. Last term's big Y, little d, there. And then we do the same for this one. First, big B, big D, actually there. So we have all of the different genotypes mapped out here. And so I did the best with the color coding that I could. And here we have RB, big D, which turns out to be purple. Big R, big B, big D, little d, which is a light purple. So it turns out that we have a purple, two light purples, a blue, two light blues, an orange, two light oranges, a green, two light greens. And even though this has the purple, this has the blue, this has the orange, and that has the green, each one of these genotypes has two of the recessive Ts, in which case the pigment is never even made, so they phenotypically are white in that case.